Thanks for the introduction. It's true. I'm working in Jülich. Jülich is in Germany, between Cologne and Aachen. It is a large. Let me just take this one. It's a large research center, 5,000 people working there, a nice restaurant, but a little bit off and very flat area compared to here. And we have several neuroscience departments there, also wet labs where we do histology. I will show lots of this soon. And we also have some supercomputers in Jülich. So Michele was just mentioning Eureka, Euron, and so on. So these supercomputers are located in Jülich. In the HBP, we also have supercomputers located in other countries, Switzerland, Italy, Spain. But we work closely with the supercomputing guys in Jülich. So my own research group is located in Jülich and working with this infrastructure, but we are also very strongly now engaged in the HBP, where we actually have a similar setup. So you have certainly seen already that we have these different platforms in the HBP, and I am working in the Neuroinformatics Platform, where we build tools to explore the brain, to get in touch with data and tools. And I'm also working in the little bit less in the supercomputing platform, where we actually build data analytics processes. So in a way, we have this cooperation with supercomputing and experimental neuroscience in Jülich locally and also on a broader scale in the HBP. So within the HBP, we have a sub-project for human brain organization. That's where we acquire lots of data, try to understand the principles of the human brain, and we actually co-design the neuroinformatics platform from here together with other scientists. We provide data and tools for the neuroinformatics platform, and in the neuroinformatics platform we do software development to build this infrastructure. And then we have this high-performance computing platform in the HBP, which actually provides the data and compute infrastructure to the neuroinformatics platform. So as you've just seen in the nice presentation of Michele, you actually can use the neuroinformatics platform to access data and compute which is running on the HPC platforms. And through the neuroinformatics platform, we provide data and constraints for modeling and simulation. That is something we have just seen in the last talk. And obviously, the idea is also to get data and tools out to the people through the neuroinformatics platform.
Sorry.
Yeah?
About the external data repositories, it's like, how is that connection made? It's like, I can access those data sets from your platform? Or?
Yeah, this is, it's good that you asked. So this depends on the repository. We are not very far advanced yet with this, but the idea is if we build such repositories in such a platform, people should be able to find and interface at least to important external repositories. One of the most important obviously being the Brain Atlas. So actually this arrow has lots of levels where we can take action. The easiest level is that we teach our platform to talk to their APIs, so that you are actually able to, when you search data, you get cross-links to the Atlas, for example, or we will also build tools that you can retrieve data from the Atlas grouped or by querying parcellations that are in the HBP Atlas, for example.
But now you don't have that, it's just something you are thinking of?
It is something we are building, but it's not yet online.
Just a last question about that as well. Are you, when you think about external data repositories, are you just considering those two, or for
no more
other data sets from, I don't know, neurodegenerative diseases that sometimes they have some connectomics data as well?
There's a whole task that is actually looking at such external repositories and making design decisions. Should we interface or not? Does it make sense to mirror the data? Do their policies actually allow us to mirror the data? Does it make sense at all to mirror the data? So people are really making design decisions there. It's a dedicated task. I'm not working myself on this task. And it's, as I said, we have not yet put anything out in that respect. I have a few slides that show particular examples later on about how such interfaces could, what they could look like. But for sure, this is an important aspect. So the idea in my talk is that I show you some human brain data that we have within the HBP at different levels. Then I try to explain what we think what an atlas is, what it should be like, and how we think that people use atlases. And also how the infrastructure that we are building should support such an atlas. And some bit more side topics, bit more advanced, how we can actually relate different data sets across levels, meaning different scales or different modalities. And I also like at the end of the talk to show you a little bit how we use machine learning to build better atlases in the future. So this is more the research we are doing. And if you have questions at any time, just ask. So we're not going so much into hands-on as we did in the previous talk, because the things that I'm showing are in development. They are not yet open. But I try to show you examples, movies, and we also see some tools live. So let's have a look at human data. Usually this slide is often around showing the complexity of human data at different levels, so that we have data really from the molecular level, nanoscale, to the level of neuronal morphologies, cellular architecture, up to the macro scale of the human brain. Can you see? And then even on the very top, we are dealing with such high-level aspects as like ethics and so on. For my talk, I consider this a little bit too broad and too much. So I try to have a look in the middle of this, focus a little bit more in the middle of this to give you some understanding. So when we consider human data at different levels, we may start at what we have already seen, single cell morphologies. If we go higher into scale, we can look at individual cell bodies, distributions of cell bodies across whole brain sections, or even whole brains in 3D. And then data at the macro scale, so where we really see whole brain images measured in vivo, lots of them in large cohort studies. This is where MRI imaging plays an important role. And then clearly we have other modalities. So in vivo imaging already, we can look at DDI data, so we see the connectivity in the brain, not the cellular architecture or the anatomical structure itself. We can also image the fiber architecture and the connectivity at higher resolutions from post-mortem imaging, for example, through polarized light imaging, or going deeper with modalities like two-photon imaging. So that means a very important aspect that we have to deal with is data on different scales, meaning different spatial resolutions. And another important dimension are the modalities. So even if we look at different scales, we may have different modalities. In this example, the cellular architecture and the fiber architecture. And there are obviously more modalities that we have to deal with. So I think you have seen talks dealing with this level of cellular morphologies, where actually 2D and even 3D cellular morphologies are extracted from imagery. Here we see work of Hype Mansfelder and Idan Segev in the HBP, where they actually retrieve two-dimensional morphologies from histological sections. You can also retrieve 3D morphologies from electron microscopy. Going up a bit higher in the scale, we deal with imaging of whole brain sections from post-mortem human brains. I'm not sure if you have ever seen this, so this is something that we do a lot in Jülich. I will show you some images. So what we do a lot is really fix post-mortem human brains by embedding them in paraffin or freezing them. It depends on the modality that we're dealing with. And then they are cut in very, very thin sections. This can be in the range of 5 micrometer up to 60, 70 micrometer. It depends on what we want to do with these sections. And we usually end up with thousands of tissue sections that we mount on glass plates and then put into different imaging devices. Yeah.
How long is the time that the brain was unfixed before it got into format? I assume format.
This depends on the brain and on the project, but this is really a little bit beyond my knowledge. So this, yeah.
This lag, this time lag, is crucial for any histology that you will do afterwards.
Yes, it is.
From death to fixation, this varies from a few hours to days,
Yeah.
usually in anatomy pathology, and crucially influences the whole thing.
Yeah, it's true.
So others, processes, neuron cell sizes, because cells are going to die immediately after the person dies in human. And so if you wait, it's valid that people do this, but you always need to know how long this brain was unfixed when you look at it.
This information is something that we take down and also put into the metadata that comes with the data. And this really differs from case to case. So we do these projects regularly, and it really depends. We have fresh brains. We also deal with sections that have been cut many years ago, and so this is really different from case to case. And you not always have an influence on that. So usually when yeah. I agree.
Sorry, also, but so this material, of course, is incredibly valuable.
Yeah.
And you get so many sections out of that. Do they all go into this type of project, or do you also have something like a brain bank, biopsy bank, where researchers could get single sections from those brains for their projects?
The idea is to provide the data to the public. And individual sections are already shared with some people, and the problem is, if you want to share all this data, you really need a large infrastructure, because it's a lot of storage required and a lot of network traffic that this causes. So this is actually why we are so active in the Human Brain Project, because we want to push that process of making access to this kind of data easier to the people.
So it's more providing data than providing material for finishing.
You mean providing access to the physical sections? No. We are usually dealing more with providing the digital data. So the sections themselves, this is actually a topic in itself, to store them in appropriate rooms with appropriate humidity and all of this. But we are not really giving the tissue away. No.
So it's not like NIH brain bank or something?
No. So just to show you this little movie again. So here's a woman who works in a lab, and here you can really see this process. This is a paraffin-embedded brain, and she's cutting the sections very carefully and bringing the tissue onto a glass slide. And then it's really very carefully, they try to make it flat with a little bit of water, get it onto the glass slide without damaging it. This is then used for different imaging modalities. And here you can see a few of them. On the left side, you see an example of a hemisphere that has been stained for cell bodies with the very classical staining, and then imaged under a light microscope. So you actually have get an impression of the distribution of neurons in general in the tissue. And this right hemisphere has been imaged using polarized light imaging. I will show on the next slides how that works. And here you basically get as an information a measurement of the direction that the nerve fibers take through the tissue. I will also show examples for this later on. This is a section for receptor architecture that's coming from autoradiography. Okay. So this is polarized light imaging. Shows us the structure. I explain a little bit how that works. So here you can see a brain section, and on the left you see the basic principle. So the idea is that you put the tissue under a light source, and the light is linearly polarized, which basically means that the waves, the light waves, are changed so that they only have variations in one plane in space, so to say. It's the same principle that you have in the cinema. If you have this polarizer in 3D cinema, these polarizer glasses, it's the same principle. There's also a polarization filter, different one in each of your glasses. It's sent through the tissue, and then basically it's measured again. And the idea behind is actually that especially the nerve fibers tend to systematically change the behavior, how the polarized light is influenced when it goes through the tissue in a systematic way. And then we change the angle that the light is polarized here and measure it again and again. So for each angle that we polarize the light, we get a new image. And the interesting information is basically how at one pixel position the intensity of the light is changing in dependence of the polarization angle. So if I run this little movie, you can basically see if we measure in different angles and you look at one position, we basically get a change in the color. And this change depends mainly on the orientation of the fibers going through, because the fibers have a lipid around them, and this has physical properties that change the polarized light. So here are basically two examples. The blue one, here's a lot of change, significant changes. So we get strong amplitudes if we look at the signal along different polarization angles. And here's an area where the changes are not very strong. So the amplitude that we see in the intensity changes is lower. And from this information, from this frequency, we can basically derive through some calculus the fiber direction and inclination angles. And this is done in Jülich by the group of Markus Axer. He's the expert for this. So here's again a little image. So for each orientation angle of the polarized light, we get one image. And if we look at the same pixel in these different images, we basically measure a sinus-like curve along these different rotation angles, and from this curve we derive properties that give us this information about the fiber orientations. And in practice, it really looks like this. People put the glass slide with the tissue on this. In this case, it's a large area polarimeter, so we get a one-shot image for the whole brain section. It gives us something in the order of 60 micron resolution, and really one image already for the whole section. And here you can really you just could see how these rotation angles are put into place. And for the very high resolutions, we can do this under a microscope, but then we measure different tiles in the image that we have to stitch together into a large image. And as you can read here, for the microscope resolution in the order of 1 micron, these are really huge file sizes, and it takes hours to do the measurement for each section. So polarized light imaging is really, really helpful to bridge the scales, indeed. So you are probably aware that we can derive 3D structural connectivity from MRI imaging through diffusion tensor imaging. So here you can compute fiber tractography on the macro scale in vivo for human brain in 3D, but this is very coarse resolution in the order of a millimeter. And on the very small scale, like fluorescence imaging to photomicroscopy, we get very precise measurements of single axons, but only locally. And with polarized light imaging, we are actually able to image post-mortem the fiber architecture for the whole brain in the range of a few micrometers to 60, 70 micrometers. So this can actually bridge these scales and really bring us from the macro scale in vivo world to the single axon world that we can measure with, for example, two-photon imaging. And this is something that we would like to leverage with the HBP Human Atlas. We would like people to be able to explore this, to dive into these different scales and actually to navigate in this data by making use of these relationships. I have to speed up a little bit. So what we also do is we try to stack these two-dimensional microscopic images together into 3D volumes. So this is one of our major goals, to really provide whole brain volumes that are consistent at this microscopic resolution. And for one brain, we already have successfully done so. This is the so-called big brain, which is available online for free, where we actually registered more than 7,000 histological sections at 20 micron resolution into a 20 micron whole brain model. We can have a look at it later on live. So here you basically have this information in 3D in the whole brain context. And we also provide maps on this scale. So for the big brain, already you can have a gray and white meta segmentation at a very high resolution. And we start providing now individual maps of different areas in this brain for the public. So now we are at a 20 micron resolution, and going higher we are in MR imaging, where we have where we can also measure fiber architecture and anatomical detail, and also obviously we have all these functional imaging aspects where we can measure activity in different brain areas under different stimuli. So some examples for in vivo imaging in the Human Brain Project. One large cohort that we have in here is from Neurospin in Spain, where we have high quality in vivo measurements of around 80 subjects, and for each of them derive the individual bundles from DDI imaging, short white matter bundles, long white matter bundles. So really from tractography algorithms for different individuals, we have the approximate fiber tracts, and from these we build average maps. I will show you some results later on from this, and this data will be accessible through the Human Brain Atlas. Also in France, Bertrand Thirion has now a very nice data set in functional imaging. This is only 12 subjects, but very high quality fMRI measurements with many different acquisitions and stimuli for not only the ones we show here, but hundreds of different stimuli for these 12 subjects in very high quality. And as you know, we can not only derive structural connectivity from this neuroimaging data, but also functional connectivity, for example, in resting state fMRI, where people are really doing nothing and the activity is measured over time. You can actually also get connectivity information by analyzing the correlations of different areas being activated at the same time, basically. So here you get also functional connectivity information. So these were examples of the data that is around. I did not talk too much about very high resolution data, fluorescence imaging, and so on. It was really more from the micron scale to the macro scale. So what is an atlas? There's a paper from a few years ago where an international consortium of bright people met, and they really discussed what are atlases of the human brain, how should they interact. And a core understanding that people have is basically that an atlas should actually provide a template and a probability distribution and a set of labels. Now this is a bit abstract probably, so let us show an example. So for an atlas, we basically assume that we have a template space. So this is usually an image showing a contrast and a definition of a coordinate system that fits this image. And the battery seems to be a bit low here. And then we usually assume that an atlas contains a map given in that space that shows different areas in that space. And the map shown here is deterministic, so at each point in the space you have a unique label to which area it belongs. But in general we assume atlases to be probabilistic. So it can be or should be that this is basically a probability distribution where you have data measured across many subjects or many measurements, and you basically give a probabilistic information. How likely is it that this position in the space belongs to this and that area? And finally, we need a set of names, a vocabulary or a nomenclature, however we call it, that tells us for these different areas what they are, and ideally how they relate also to other atlases, if this is at all possible. So to show you some examples, for sure this is not a comprehensive selection. In the Human Brain Project we have an atlas for an infant brain from Ghislaine Dehaene in France. Here's an image of an initial version of a white matter bundle atlas, which is actually derived from the structural connectivity in the Archi database. So if I go back, we have seen that from these data sets. So from these individual white matter bundles, people actually looked at the statistics across all the individuals and derived a map. And behind the map that you can see here, there is also probability. So this is only a summary map that shows you the most likely label assigned to a voxel in the white matter for the different bundles. But in the background, this is a probability distribution. So for each voxel, you basically get a probability how likely it was that this voxel was part of this structure in this cohort. And on the right, this is the Ubrain Cytoarchitectonic atlas that we built in Jülich that shows you a parcellation into cytoarchitectonic areas. We look a bit deeper into how this is done now. Do you have any questions so far? Okay. So for the right one, the Cytoarchitectonic atlas, let's have a look how this is done. What people are doing to find these different areas is they look into these histological sections stained for cell bodies, as we have seen a few minutes before, these 1 micron sections where we see the different cell bodies. And if you look into cortical areas, you basically see these neuronal layers 1 to 6, this columnar structure, and in different areas of the brain, these are 2 visual areas, you can actually see that the distribution of cells in the different layers is significantly different. And what people do for decades already is they pull out these histological sections, work on one area, and really in different sections try to find these significant transitions between areas and map them in different subjects and different sections for sure. And how that works is basically so that it's not arbitrary. They do a segmentation of the cell bodies, and then they basically look along such profiles in the non-overlapping profiles across the cortex. They look how the statistics of these segmentations change. I have a little animation of this. So if we have here one example, this is extracted from such an image. And we have proton area 11 and the frontal pole here to the right. We actually assume that the profile of the segmented cells, so the information of the along the cortex in this direction, along these profiles, is different for these different areas. So here you can see a peak more to the left, which I guess refers to these layers here with a more dense cell packing, and here in this example, the peak is more to the right, more in layer 6 than layer 5. And the question is now, how can you measure this? And the idea is really to measure the correlation of neighboring profiles along the cortex and look for significant peaks to verify such a hypothesis that there is a border between areas. So this has been done for decades. This method has been proposed by Schleicher. So the mapping has been done for decades, and this method by Schleicher to somehow find these areas in an observer-independent way has been introduced in the 90s. And this is the basis actually for these cytoarchitectonic maps. And this is very tedious work. You have to do it in many histological sections and in different brains. So usually for one area, a PhD student is working for some years to map this one area. It's a really tedious work to do. And people typically do this in 10 or more post-mortem brains. So obviously we don't want to do it in one brain only. This would be more or less arbitrary. It's done independently in different brains, in each of these brains in many different sections. And then we are interested in the statistics of these areas in the different brains. So we do this in many different brains. And then to actually arrive at this probabilistic parcellation, this probabilistic atlas, we have to merge or bring all these different brains into the same coordinate system, which is very difficult, to actually get a statistic at every voxel in the same space, how likely it is to see that this voxel lies in this particular area. And you can see this in this slide. So here we show only 3 of the 10 brains where these areas have been mapped independently. And the idea is to merge these 3 or 10 into one reference space, which is the MNI-Collin space that we use here. And then you get this probability map, which basically tells you red means this voxel has been mapped as this area in all of the brains. And blue means only in a few of the brains. That's basically what it's saying. So it's probabilistic information. And this is only one example. We have different atlases, as I've shown, also for the white matter bundles, and this is done in a similar manner, not exactly the same. So this is one example how we can do that. So how do people use these atlases? The most obvious use of such an atlas is to explore the anatomical and functional properties of the different brain regions. So you want to look at it and see this information and explore it in 3D and navigate it. And maybe also use this to find more data. And we have a little prototype, rather simple, but and people certainly want to, besides only navigating in 3D, they want to be able to cross the scales interactively. So you want to zoom into the data and then at a high resolution probably find another data set measured at the same position at higher resolution showing the same structure. This is a picture of a subthalamic nucleus. And here we have a data set where we extracted the 3D cellular distribution in the subthalamic nucleus. And it also shows a partition of this distribution. So we want to provide people the possibility to cross the scales interactively, to really navigate the whole brain, go into a region, find higher resolution data, more specific data, and go deeper and fluently explore it and find the data that they're interested in for their studies, and then be able to use that data for their analysis. Besides simply exploring the data, an important use case for atlases is in vivo imaging. So what people already do today with these atlases is they use them to, for example, interpret functional MRI results. So if you have activations in the brain, you would like to know in which areas are these activations, or are they spread across different areas? What is the percentage of this activation in this and that area? This is something we can provide with these atlases. It's already possible today with the SPM anatomy toolbox that has been implemented by Simon Eickhoff, but this is an important use case for atlases in general. And then even going deeper, you can use atlases to study, for example, differential gene expressions in the different areas. This is something that we have already built and would like to make it available in the Brain Atlas. So you could navigate into different parcellations of the brain, select areas, and then retrieve different gene expressions, for example, from the Allen Brain Atlas by using the API right away, and then find genes that have a strong differential expression in these two areas. So they are expressed significantly different in these brains. And you can also do this for other modalities. Use the atlas to group the underlying data and run differential analysis. And another important use case, this is connecting to the talk of Michele. We have, especially in the Human Brain Project, this is very important. We have people building models of the brain. So for example, we are working here with people from the group of Gustavo Deco. They have whole brain network models of human brain activity, and they would like to make use of atlas data. So what we are basically doing, this is really not working. Through the atlas, we provide them constraints based on experimental tractography from DTI, cytoarchitecture, and the reference atlas that they can choose. So this gives them constraints for their model, how many cells are in the different areas, what is a typical cell distribution, how is this area connected to other areas in the brain. This is information that they use to trigger their model. But that's not all, because they also get results from their model. The model basically predicts resting state connectivity. And so from the atlas, we also compute an empirical connectivity matrix for the different brain areas based on functional MRI. For example, the functional MRI datasets of Bertrand-Thirion that I've shown. And the modelers can then use this connectivity matrix to validate their model. So here we get a connection from the experimental data to the models through the atlas. And we are actually building infrastructure to facilitate this. Today, people are really sending around the data. So they, like the lab of Jeff Marjan is sending this computed connectivity matrix by email to these people, but we would like to provide such things as a service so that modelers can go to the platform, can actually select a parcellation, select different sets of underlying experimental data, and then based on these selections create a custom constraints and also custom connectivity matrix to use that for their models without contacting the people in the labs. So how can we support this with infrastructure? As I said, the Neuroinformatics Platform in the HBP is, amongst other things, will host these atlases. And the idea is that you can get access to well-curated reference atlases, to lots of this heterogeneous human brain data, which you can find through the semantics by a metadata search, but also much of it you can find it by a spatial search, by navigating these different spaces. And it can also provide access to external repositories. And the infrastructure should allow you to discover this data, to register new data, get your own data into the system. This is an important aspect. And it also allows you to run analysis with the data. Just in a similar fashion as you have seen for the brain simulation platform, you will be able to run analysis of experimental data in the collaboratory. So finding data is obvious. There is a metadata search engine where you can basically say I'm looking for cytoarchitectonic parcellation. I'm looking for a neuron model, whatever, and you will find this data. This search is under reconstruction currently. For the navigation, we are now working very hard to get this out. We want people to really be able online on the web to see even these high resolution data sets. So it's today possible to download an MRI brain for you or such a macro scale 1 millimeter resolution atlas and navigate it in 3D on your computer. But if you want to navigate a data set like the big brain that is 20 micron isotropic, this data set has a size of 1 terabyte. So you cannot easily download it to your computer to navigate it. Your computer cannot even load this into memory. Usually you won't have 1 terabyte of main memory on your machine. So, and you don't want to download it because it will take hours. So what you want to do is you want to explore it on the web just like you do explore the Earth in Google Maps. The idea is, so this is still a mock-up, this is not yet available. The idea is now that we embed this into a more complex viewer where you can actually select species, templates, and parcellations. So these are the 3 components of reference atlases that I've mentioned. You can select them, then you can see the names of the areas here. You can select them, and the viewer will go to this area, navigate to this area, and you should also be able to select a range of reasonable tools. For example, similar to what I've shown in this papaya viewer some slides ago, that you can interactively search for more data according to the selected regions. So this is work in progress. We are building it already, but this is not yet available on the web. So how does infrastructure support such an atlas? One obvious aspect is the collaboratory. You have seen it for the brain simulation, and we will also have interfaces in Jupyter notebooks for the collaboratory to access the atlas. So this is also not yet there, but the idea is that people working in simulation can directly access the atlas, make queries in the atlas from such a Python notebook, and then use that data for simulation, but also run data analysis in the collaboratory in a similar manner as you have seen it for the simulation. And another aspect that makes use of the high performance computing platform, this is an example for neuroimaging, is that we make these computing resources available for people to analyze huge data sets. So we are working to register lots of different neuroimaging data in the Neuroinformatics Platform. Much of it, we make it available really as data on the backend, so we have it stored in the background. And then, I don't know, is anybody here working in neuroimaging? Okay, so you know that you usually have these pipelines. You process all these in vivo data sets. There are these established pipelines, FSL or whatever, that you use to pre-process the data, and then on the pre-processed data you run a group analysis or whatever. And if you want to use really large cohorts, it's getting difficult to do it. You need a cluster computer or whatever. If you want to do it on the extreme case, UK biobank data or whatever, it's not easily possible to compute all this pre-processing yourself on all this data, especially if you want to try out different settings for the pre-processing and so on. So what we want to provide is actually workflows installed on a supercomputing to do such pre-processing with different settings on these different data sets. Be able to configure such workflows, run them on the supercomputers, and share the derived data, so that you would be able to compute group analysis on large selections of neuroimaging data with different settings, and provide that to your colleagues or to the public through the platform. Things that you couldn't easily compute on your own machine or in your own lab if you don't have access to large computers. So this really uses both of the platforms in common. And more simple aspects that where we need the platform to provide tools for atlasing is we want to get new data in. And I've shown you this video where we actually navigate into the big brain, and then we find this subcortical structure, and suddenly we would find a higher resolution data set that corresponds to this structure, and then go deeper. To realize such things, we actually need people to come with their own data and register them to the system, and also be able to say where this is. So people might have an analysis of a partial volume of a brain at very high resolution, and then do not only want to provide it to the public together with the metadata, but they also want to make an alignment to the template spaces that we have, so that it is approximately aligned, and you can also find it by spatial searches. And for this to work, you actually have to provide people tools to register their data to anchor their data spatially to the reference atlases. And I think on Monday, Trygve Lergaard has shown some of such tools for rodent atlases, I assume. So assume that you have a local 3D reconstruction of the subthalamic nucleus as a very high resolution, and you want to align this with the big brain, which is at 20 micron. This could be at 1 micron. How would you do that? I think that Trygve has probably shown you this Quickney tool. Did he? I'm pretty sure. Where you can have a single section and anchor this section spatially to a 3D reference atlas in the red brain. I thought that he has shown it. So here's a little movie of how that works. You have this section here, and here you see the on the right, you see the 3 sections of a reference volume. This is now the big brain. And then you can use this tool actually to find for this one image how it is aligned in the 3D space. So you can find this 3D transformation that maps this one image into the 3D space. Now, if you want to do this with volumes, with partial volumes, as I've shown before, and you want to align this to a high-resolution template like the big brain, this whole stuff has to be computed online. So you have to somehow upload your partial volume, and then through a web browser remotely do this alignment in 3D. And we are also working on tools to enable this. This is a view of a current prototype that we have. We are already using it, where you really have 2 3-planar views of a reference volume and of the volume that you want to align. And you can add 3D landmarks interactively and say this landmark is here, this is here, and it will find the 3D orientation of your structure in this template space. So you are able to submit 3D data to the system and do a spatial anchoring in 3D yourself. This is a crucial aspect to basically get people in to enrich the whole system with new data. So we have a bit of time left. I would like to show you some aspects that we use in between all of what I've shown for now. One very important aspect is image registration. Who of you worked with image registration already, or knows what image registration is? 1, 2. So not too many. So I try to give you a little introduction to image registration, because in almost everything I've shown here, we have to rely on image registration. You have seen, for example, that we have these different mappings in 10 different brains, and then we fuse them into a common reference space to get this probability information in the same space. To do this, we actually have to take these 3D volumes and deform them to be in the same coordinate space, because they are not naturally in the same space. This is where we need image registration. It gets a little bit simpler if you think about 2 images. This is a coronal section of the human brain. This is the next section of the same human brain, so it's a bit different. And it's imaged under a microscope, so it's probably a little bit rotated, a little bit moved to the left, to the right. So it's not, if you superimpose these 2, they're not exactly the same. This is here stolen from the manual of Elastics. It's an open source image registration toolkit. They have a very nice picture. So if you do this in 2 directions, now I only moved the image in the X direction. I can also move it in the Y direction, so I get a 2-dimensional space. And then you can basically see that, and then for each of these 2D offsets, I measure the similarity. You can see that I get somehow a landscape, like a mountain, with all the difference values. And image registration will look for a good minimum value in this mountain and assume that this minimum value gives you the best transformation of the image to fit the other one. That's the idea of image registration. And obviously, only moving the image around is not sufficient. So if we have a 3D volume of a brain, it will not, and another one, it will not be sufficient to just move it around in the three coordinate directions to find optimal alignment. Instead, we have to use more complex deformations. Here you see some different ones. This would be only moving it around, but you would also like to rotate it and maybe shear it. And in the most complex case, and for human brains, this is what you have to do, a nonlinear deformation. So really deforming the brain and searching the best nonlinear deformation of the brain that fits the other one. So here you can see a toy example. We have a grid. This grid models a deformation, and by moving these red points around, you deform the tissue. Here you can see random deformations, but you can see that this is a simple model that allows you to deform the image. And you can assume, just similar to moving the image into the different directions, that you can also look for the best movements of these control points to deform the image so that it fits very nicely. And this is the idea that we used, for example, to build a big brain. We have this stack of all these 7,000 sections measured in the histology, and we deformed every single one of them to match its neighbor as good as possible. And we had to deal with problems like this. In the histology, sometimes tissue is ripped off, so this, I'm going a little bit through this, but the idea is really that you align all these consecutive sections to get a 3D model. So this uses image registration. And for the atlas, if we want to relate information across the different atlases, we need to do something very similar. So as I said, we will have different atlases, and then each atlas may be defined in a different template space. So if some people provide data that is aligned to Atlas A, and some other people are browsing Atlas B, they need such a transformation to be able to access the information in the other atlas if they want to access it spatially and not only through their metadata. So we need transformations between the different template spaces. And here we are really implementing such nonlinear deformation algorithms into the platform and also storing very precise deformations for the default template spaces that we have on the platform so that you have them available. So you can actually, in one atlas, select a point, and the system can immediately say where this point should be in another atlas. And the alignment that we use is not only such a nonlinear one, it's one that also uses patterns of sulci in the brain to stabilize this deformation. We don't have the time to go through all this anymore. Just as a final remark, because I promised it, as a very last thing, I want to mention that we use machine learning methods and we try to improve the mapping that we do to build these different atlases to actually be statistically more stable. So we actually want to do these mappings, these delineations of different areas in more brains, faster, with more data, and this cannot be done by hand anymore, by atlases. So what we try to do currently is we want to investigate, neural networks as a machine learning model. I don't know, is anybody familiar with machine learning here? So quite some. So here we use something that's called deep learning. So you basically have an artificial model like a neural network, and you train this model by showing it examples of input and output data to actually replicate this, to given some input data, show you the output data. And here what we show to this model are these mappings of the neuroscientific experts that I've shown you previously, so people mapping the brain, and we show the network different patches of such images, and also the area that it had been assigned to by these experts. And then we investigated if these networks are able to learn this behavior, and for new data and for new sections make their own decision to which area this belongs. It's a very recent work in our lab, and it's quite interesting, so I skipped the details. But we have trained networks that are able, given a section that they have never seen before, to basically make a guess about the different areas, where these different areas are, on a pixel basis. So on each pixel of the image, they can make a guess how likely is it that this area is V1, V2, whatever. And this worked quite well already, although it is a very difficult problem because the number of possible different images you can get in these microscopy images is really, really, really huge. So the network has still learned to distinguish them, and the visual areas are the simpler ones. So here we should expect that a good model is able to do it, and it does. So we have currently an artificial neural network that tries to mimic the behavior of the neuroscientists when mapping the brain. And we are investigating this further. So currently we use this network to predict in new sections these different areas. And in parallel, we have a neuroscientist doing it. And then we meet and we look at the differences. So we try to get an understanding, what did the network learn? What makes an area compared to what the neuroscientist says. And as you have seen, the neuroscientists use these cortical profiles, and they look how the cell packing density changes across the cortex, but the neural network doesn't do that. It just looks at the images, and it can derive any information from this to make a distinction between the different areas. And this is very interesting. So we have had examples where the networks spotted errors that the neuroscientists did. And we have also found areas where it's different, but it's very interesting that it's different. So where we get a feedback is this traditional method of looking at the profiles, is it appropriate or not? So this is very interesting because we have an independent, very fast method that does a similar work on lots of data. So we can use it to verify the mappings. We can use it to produce much more mappings in much more different brains, so we get more statistics. And maybe we can even derive new methods to distinguish different structural areas, new features from this. So this is a very promising new work that we are doing, and the first thing that we are now splitting off this work is actually we use these networks to speed up the work of the neuroscientist. So while the neuroscientist is mapping, the network is trying to learn already what he's doing and predict this in other sections, so that it's that the neuroscientist doesn't have to do it in all the sections, but the network already does some sections by learning in the same brain. So we are already a few minutes late. I would have had another example, but maybe we stop at that point. Are there any more questions? So otherwise, thanks a lot for your attention.